Okay, thank you. I want to first thank the summit organizers for inviting me to give this talk. My name is David Torres, and again, the title of my talk is Modeling the Influence of Mites on Honeybee Populations. This was previously published as a special issue of Honeybee Health in 2020 in Veterinary Sciences. Also like to acknowledge my co-author, Nicholas Torres. So why study honeybees? Well, honeybees are involved in honey and beeswax production. You may not know, but honey does not need to be refrigerated and does not spoil. This is due to the fact that it's hygroscopic, which means it extracts moisture from the air. In fact, 3,000-year-old edible honey has been found in Egyptian tombs by archaeologists. Darker honey has more antioxidant properties, for example, honey made from buckwheat, than lighter honey. The way it works is that nectar is processed in bee stomachs. Enzymes are added when passed between bees, and this process breaks the nectar down into simple sugars. It is deposited in hexagonal cells and fanned by honeybee wings to reduce its water concentration. However, I don't want to linger on just honey and beeswax production because it pales in comparison to a more important role honeybees play to our economy and agriculture, specifically that of pollination. In fact, it could be estimated that pollination is 50 times more economically important compared to honey and beeswax production. So the crops that are almost completely dependent on honeybees for pollination include almonds, apples, avocados, blueberries, cranberries, and so on, up through pumpkin, squash, and sunflowers. And the bee pollination of agricultural crops accounts for a third of the United States diet. In terms of the outline of this talk, I first will discuss the honeybee, the insect, its caste system, and division of labor. Then I will move on to discuss threats to honeybee survival, the mathematical model which we developed, results and sensitivity studies from the model, and finally I will conclude. So the honeybee belongs to the class Insecta. Bees are found in every continent except Antarctica. And out of the 25,000 species of bees, only 7 produce and store honey. Honeybee colonies employ a caste system based on classes, and bees move through the different classes as they age. So all bees start out as eggs and remain as eggs for 3 days. The queen bee may lay up to about 2,000 eggs in one day. Then they move on to become larvae, which look like grub-like white sausages, where they remain there for 5 to 6 days. Then they graduate and become pupae, where they are sealed in these hexagonal honeycomb cells, and they develop as pupae, and after 12 days become worker bees, the familiar worker bees which we're familiar with. They can remain worker bees as hive bees for 21 days, tending to the larvae and processing nectar. Some of them become the drones, which are the male bees, but there are only a few hundred drones in a honeybee colony. And their really only function is to fertilize the queen bee. After the 21 days during the summer, they become foragers, and their lifespan is limited to maybe 14 days, it depends. And it's a hazardous occupation. I mean, they're subject to the elements and other pests. So honeybee colony can contain up to 50,000 hive bees. So here we see a picture of the hive bees tending to the larvae. And some in the honeycomb cells, you can maybe see the smaller ones, eggs. And some of these are capped. So you might see some of these honeycombs are capped, and that's where the pupae are developing. In this next slide, we see a queen bee, the drone bee, which are usually larger than the worker bees, the hive bees, which are more predominant in a honeybee colony. And this can be found at this website. Now, you may know that pheromones regulate bee activity. There's the alarm pheromone, which is released when the bee stings. There's the brood or larvae pheromone, which inhibits ovarian development in the hive bee. And there's ethyl oleate, which is released by foraging bees to slow the maturation of hive bees. So ethyl oleate is a self-regulating mechanism to restore the balance or ratio of hive to foraging bees. And one study showed that honeybees leave scent marks on flowers that repels other bees, possibly informing bees that the flower has been visited. In the summer and the winter, in the summer, foraging bees gather nectar for the hive and indirectly pollinate a wide diversity of plants. In the winter, adult honeybees live for months in the hive, shivering to generate heat that preserves the hive during the winter temperatures. The metabolism slows, and honeybees are much less active. A few interesting bee facts. Bees actually can see in the ultraviolet range. They tend to seek out flowers of the same species, and they have a dance they perform called the waggle dance, which is used to inform other bees of nectar sources. So honeybees have been under attack recently. They've been declining, and this is due to many threats. These threats include the parasitic mite, known as the Varroa destructor, and this mite may have eliminated most wild bee honey colonies in the mid-1990s, and it's associated with viruses. So there are 20 known viruses. The most prominent ones are the Israeli acute paralysis virus and deformed wing virus. There's also the protozoan Nosema ceranae. Pesticides, particularly the neonicotinoid insecticide. Habitat loss, environmental stresses, bee management practices, tracheal mites, foul brood, fungus, wax moths, wildlife, and invasive plant species that reduce nectar-producing plants. In this talk, we're going to be focusing on the Varroa destructor mite. So these are these small 8-legged external parasites that reproduce in the actual honeycomb. They reproduce in the honeycomb, but in the phoretic state, they remain attached to the bee, and they feed on the body fat tissue of adult bees. They can create open wounds, which make bees vulnerable to additional pathogens, and some mite populations have become immune to miticides. So let me move on to discuss some mathematical models. It's difficult sometimes to observe all the different dynamics within a honeybee colony, so people have used mathematical models beginning in 1989 with de Grande, Hoffman, and then moving on to Martin, Schmichtel, and Crailsham, Corey, and Beecher. And some models incorporate the mites in the model or incorporate how the mites affect the honeybee colony. So what's unique about our model? Well, we use coupled differential equations to model the honeybee colony. Our equations track the number of bees in each day of all the castes, the egg, the larvae, the pupae, the worker bee, and the forager bee. The model accounts for the drones. It turns out the drones are important in regard to the mites, even though they make up a small percentage of the population of the honeybee colony. We do account for the larvae requirements for hive bees, or if there are insufficient hive bees, that reduces the survival of the larvae. We account for pheromone effects and the maturation changes it causes in the hive bees. We do have healthy and infested classes within each caste. So the healthy ones do not have the mites, and the infested classes do have the mites attached. As far as the mites, we do look at the phoretic stage when they're attached to the hive and forager bees, and the reproductive phase when they're within the capped cells. We do also allow for grooming, in which bees remove the mites. So this is a differential equation we'll use. Here, BI stands for the number of bees that are I days old. And here we see the rate of change of BI with respect to time here. This term here, this S I minus 1, B I minus 1 minus BI accounts for the natural aging of bees. The IAI can affect the maturation rate of bees due to pheromones. This term here accounts for the fact that bees can become infested with mites, and this term is the grooming term. And we can use different survival rates for each caste, for the eggs, the larvae, and so forth. The survival rate of larvae is reduced if there are insufficient hive bees. So we look at the ratio of the hive bees to the larvae, and that changes the survival rate. So here are some more mortality and survival rates we're using. These are daily ones. The survival rate is 1 minus the mortality rate. This was based on Schmichtel in 2007, who based rates on experimental data from Sakagami and Fukuda. So you can see it changes, the mortality of the bees changes depending on the caste they're in, and it's highest in the forager caste here. Here's a cartoon or illustration of the bee model. So we track the number of bees that are I days old. So you can think of these as bins or boxes. And during a day, a bee will move from one box to the next box. You know, this is the number of bees that are I days old. This is the number of bees that are I plus 1 days old. But not all bees make it from one box to the other. You can see that we multiply by this factor SI, which is survival rate, the daily survival rate, as we move from one box to the other. We do have the healthy class and the infested class, and we can use a different survival rate in the infested class. And then there's migration from the healthy class to the infested class here due to infestation, and then movement from the infested class to the healthy class due to grooming. As far as the percentage of eggs that become drones, we use this equation from Martin in 2001. It depends on the day length and the number of hive bees. So this is actual simulation from the model. We see that the percentage of eggs that become drones peaks around 4% here. And this is for a 2-year simulation. So here on the x-axis, I'm plotting the number of days. So about 720 days, say, in a 2-year, or 730 days in a 2-year cycle. In regards to the mite and the model assumptions, we assume that the daughter mite offspring are produced at the rate of 1.5 for the worker pupae and 2.6 for the drone pupae. So a foundress mite will enter into one of the honeycomb capped cells, or before it's capped, and then after the 12 days, 1.5 additional mites will be produced for that foundress mite in a worker pupae, and 2.6 mites will be produced for the drone pupae. This is partially due to the fact that the drone pupae remain there for 14 days compared to the worker pupae, which they remain in that phase for 12 days. The model distributes 75% of total mites to larvae cells before they're capped. Mite do have a preference for the drone brood or the drone pupae. And the mite and pupae survival is reduced in cells that are infested with more than one foundress mite. So sometimes more than one mite enters one of these honeycomb cells before it's capped. And that causes a decrease in survival, not only for the mites, but also for the bees themselves. With both those survival rates are decreased. And we assume the mite lifespan is 27 days. So I'm going to show you simulation next. And these are some of the simulation parameters we used. We started with about 8,000 hive bees in February 1st. This was the maximum egg laying rate of the queen. So the queen doesn't lay at this maximum egg rate all the time, but it kind of ramps up to this number and then decreases late in the season. We have an ideal hive bee to brood ratio, an ideal hive bee to forager ratio, which is used in determining how pheromones regulate the activity in the hive. The latitude is used to determine the day length. We use a time set of 0.1 days. And the length of the summer was March 6th to September 17th. The initial number of mites was 200. And we have these mite daily survival rates in the summer and the winter. So we have an extra differential equation with models of mites as well. The grooming rate here was 0.05 and the transmission rate was 0.25. Those come from these terms, which in the differential equation. The grooming rate here, beta, and the grooming rate gamma, and the transmission rate beta. And then we have the reproductive rate of the founder's mite in the drone and worker brood. So this higher rate in the drone brood is actually an important factor. So my first simulation here, I'm going to basically model the different castes within a honeybee population, given those simulation parameters. Here, I show you the eggs over a 2-year span, and the increase in the eggs, the decrease during the winter, and then the increase again during the summer. These are shown as these blue circles here. The larvae are these black diamonds here. The pupae are the blue squares. And then we have the infested population or classes as well, the infested pupae, which come later in the season. And also I show the drones, but I multiply the drone numbers times 10 because they're much smaller. So the numbers here have been multiplied by 10 before I plotted them, just so they show up on the graph. In this simulation, I want to note that we assume the mites do not affect the mortality of all the other bee castes. This is just to track how the mites do without affecting the mortality. We're doing the same simulation here in this slide, but now I'm looking at the hive bees, the infested hive bees, the foragers. These are the green triangles here, the foragers, and the infested foragers, these asterisks, and the mites. You see the mites tend to increase later in the season, and so it's staggered. These sort of the peaks there are staggered, and the mites actually come, the peaks in the mite population comes later in the season. So we can also track the daily % rate of increase in the mite population here. So there's a lot of volatility early on in the season in the model, but that stabilizes. The average was about 2.6% in the first year and 1.3% in the second year. Reported % growth rates are between 2.1% and 2.5%, depending on the source. This graph shows the percentage of mites in the cap cells. So it's around 70%. The model was shooting for 75%. So we almost achieved that, but there's some volatility in the percentage of mites in the cap cells. In the next simulation, we actually allowed the mites to be harmful. So they increased the mortality of the caste. And in this simulation, the mortality rates in the infested pupae, hive, drone, and forager castes are increased by 4 times with the harmful mites. And what I'm plotting here is the difference, not just the total population, so the difference between the simulation with the harmless mites and the harmful mites. So we see that here, when I'm plotting the difference, there are more bees. So there's more pupae here, which is expected because the harmless mites do not affect mortality compared to the harmful mites. So there are more pupae when we look at the harmless simulation. We also see a slight increase in the larvae. The drones are the biggest ones. So the drones tend to be, the numbers in the drones are the highest when the harmless mites are used. But notice the scale here. The numbers overall are pretty small. The drones are the only ones that significantly get affected. The populations they do affect, or the castes they do affect, are in the hive bees. You see the numbers, the scale here are much larger, and in the forager bees here. So there's about 1,500 difference in the forager bees. So there's much less forager bees when the mites affect the mortality. And again, we see here the mites actually are increased when they are harmless. So the mites have a negative effect when they actually negative effect on themselves when they negatively affect the population. They do better when they don't decrease the mortality of the bee caste. And this actually simulation didn't proceed into the second year. The mites killed off the honeybee colony. In the next simulation here, I use 32 different grooming rates ranging from 0 to 0.15 here on the x-axis, and 32 different transmission rates here. So the transmission rate is a rate at which bees can become infested with mites. And we see, and what I'm plotting here is the maximum second-year high bee population. So when it's high, here in the yellow, the bees actually, the colony survived, okay? But it's when in the blue, the bee population actually perished. And there's a sharp line between when it perishes and when it survives. So the grooming rate makes, if bees are allowed to groom and remove these mites, that makes the colony much more resilient, as you can see here. But when the transmission rate increases, it tends to decrease the survivability of that particular honeybee colony. In this second simulation here, again, I'm plotting a different 32 different grooming rates here on the x-axis, and on the vertical axis, I'm changing the drone mite reproductive rate, the rate at which they reproduce in the drone pupae. And you can see the drone actually, you might not think the drones play an important role, but they actually do. If you increase the reproductive rates in the drone, that can make a difference between whether the colony survives and whether the colony perishes. So here I'm plotting as a color contour the maximum second-year honeybee high population. And there's a sharp line between survival and whether the colony perishes. And some farmers actually remove the drones to improve the survivability of the honeybee colony. So if you're interested in code, it can be downloaded for free at this site, this GitHub site. You can actually email me. It's written in MATLAB. It also runs in Octave, so Octave can be downloaded for free. MATLAB requires a license typically. And the 2-year simulation times there's a wide range of simulation times, ranging from less than 1/10 of a second, depending on what version of MATLAB you use, to maybe 84 seconds with Octave. So it takes longer with Octave. So in conclusion, we have presented background information on honeybees, their caste system, and threats to their survival. We've used the biology of a honeybee and the Varroa destructor mite, the caste system of the honeybee, the survival rates and pheromones to build a mathematical model. And we see that the reproductive rates of the mites, particularly in the drones, the grooming and infestation rates are important parameters which determine if the colony will survive or perish. So if you have any questions, you can email me. Here's my email address, davidtorres@nnmc.edu. So that concludes my talk.